Hello my dear friends, welcome to Live Prep. In the previous lecture, we studied about the proteins, their structures and conformations, everything we have studied. Now in this lecture, we will go with some nucleotides. So here, what are the components of each nucleotide and what are their structures? With structures, please remember. Here we are focusing very clearly on each and every structure because in the NCERT, in this particular chapter, clearly they have not mentioned, but the problem is they have directly given the names and everything. So we need to understand these structures, then only we can understand those names. That is the first thing. And the second important point is the same content, please remember, same content with structures, everything. Once again, we need to study under molecular base of inheritance chapter, because in that chapter, to understand the structure of DNA, to understand that remaining other concepts, bonds, calculations, many things are there. For all these things, this is the basic information. So every student, at least, so here in this lecture, first what I will do is I will give the individual structures of all these things in this particular lecture. In the next lecture, what I will do is I will combine this. After combining this, different names will come. At least in NCERT they gave these names, and some of the structures also they have mentioned. So what I am saying is you just try your best to understand these structures, and if possible practice the structures and try to remember them with structures. Then only you can understand the NCERT content. In this chapter they have not given good content, but in the molecular base of inheritance, first 2 pages, they have given very clear-cut information regarding more information with respect to structures. At the 3' what is there, at 5' what is there, at 1' what is there, and which one is attached to which group, which molecule is removed, what is the bond that is formed, everything they have mentioned. So at least there you need to remember everything. So my suggestion is from now itself you just start working on it so that it will become very easy for you. But this is very important and it is interlinking also. So that's why I'm spending more time. So please remember. So in this lecture, I will give these individual structures. In the next lecture, I will try to combine all these things so that nucleotides information will be completed. And after that, there is a separate lecture. There we will mainly talk about nucleic acids, and there we will talk about DNA, RNA, some basic information and some information related to structure. Likewise, we will study. So that means we are spending total 3 lectures for this. I hope you can understand how important it is. And I am trying to give you all the details because anyhow the same concept will come once again. So at that time, we don't need to spend much time because already we are covering with clarity. So that is the reason why I am going a little bit slow. In this lecture, we will try to complete each and every structure of this. With that, this lecture will be completed. Clear? And now we are going with the information. First, I will give you the explanation. That's it. Here, nothing is there. This is what the table is. I will tell you each and every point. And now we are focusing mainly on the structures. So now in this lecture, first we are starting with nucleotides. Nucleotides. Why we are talking about nucleotides? Because in the next coming lectures, we will study about nucleic acids. Nucleic acids. Please remember, in the macromolecular fraction, in the retentate, 4 organic compounds are there: proteins, nucleic acids, polysaccharides, and lipids. Already proteins we have completed, and now we are going with nucleic acids. Before directly starting with nucleic acids, what we studied, nucleic acids are polymers of nucleotides. They are the polymers of nucleotides. What is the meaning of this? Each monomer, each monomer is called as nucleotide. Each unit is called as a nucleotide. If all these are combined, then it is called as a nucleic acid. So first we will focus on one unit. So previously what we studied individual amino acid. After attachment of amino acid, protein will come. So that's how we have studied. Same way, here also first we will talk about one unit, that is called nucleotide, and then we will study about the complete molecule called nucleic acid. So that's why first we are focusing on one unit, and that one unit is called as nucleotide. So the monomer of nucleic acid is nucleotide. And what is that nucleotide? In nucleotide, again, even though it is one unit, inside this also again other components are present. What are these? So nucleotide. Each nucleotide, tide 3, you can understand this, 3. Nucleotide is having 3 components. What are these 3 components? First one is nitrogenous base. Nitrogenous base. It has 3 components. Out of these 3, the first one is nitrogenous base, and the second one is sugar. Second one is sugar, and the third one is a phosphate group. Third one is phosphate group. These are the 3 components that are present in the nucleotide. Within the nitrogenous base, again 2 types of nitrogenous bases are present. They are purines. They are purines and pyrimidines. They are purines and pyrimidines. Please observe the content carefully. And within the purines again, 2 types are present. What are those? Adenine and guanine. Adenine and guanine are the 2 purines. You need to remember each and every point. Each and every point is very important. And total 3 types of pyrimidines are present. What are those? Cytosine, uracil, and thymine. Now I will give you 2 options. What is the correct spelling? You need to tell me. THIAMINE. Second option, THYMINE. Now tell me. You are saying thymine, but what is the exact spelling? What is the exact spelling that we need to write under the pyrimidines? This is very important. Many students are making a mistake here. And the answer is THYMINE, thymine. It is the THYMINE, thymine. Very important. Spelling is very important here. Thymine. If THYMINE, thymine is pyrimidine, then what is this THIAMINE, thiamine? It is the thiamine. It is a vitamin. Thiamine is a vitamin. So please observe carefully. So THYMINE, thymine is a pyrimidine, but THIAMINE, thiamine is a vitamin. So here we are talking about the thymine as a pyrimidine. So please observe carefully. So in this way, in the nucleotide, 3 components: nitrogenous base, sugar and phosphate. Overall, if you see, nitrogenous bases are 2 types, purines and pyrimidines. Why they are called purines? Because they are having the basic purine ring. Or in simple, you can say that adenine and guanine are substituted purines. Adenine and guanine are substituted purines. So that's why we are considering them under purines. Whereas cytosine, uracil, thymine, they are having the pyrimidine ring. So since they are having the basic pyrimidine ring, they are considered as substituted pyrimidines. So in this way, nitrogenous bases are classified into 2 types, purines and pyrimidines. Within the purines, based on the structure again, adenine and guanine, 2 types are there. Adenine is indicated with A, first letter, and guanine is indicated with G, first letter. Okay. Next one, cytosine is indicated with C, first letter, and uracil is indicated with U, that is the first letter, and thymine is indicated with T, that is the first letter. This is how we are generally using these particular nitrogenous bases and their names. Now come to the sugar. And what type of sugars are there? 2 types of sugars are there. If we are talking about the RNA, what is the full form of RNA? Ribonucleic acid, ribo sugar is present. In the case of RNA, ribo sugar is present. And what is the full form of DNA? Deoxyribonucleic acid. Is that no? Deoxyribonucleic acid. So deoxyribose. It is the second carbon deoxy. So it is the 2-deoxyribose. It is a 2-deoxyribose sugar. So in this way, overall if you see, 2 types of sugars we are observing. Ribose sugar, deoxyribose. In this deoxy also, what is the carbon that is affected? At the second carbon, oxygen is removed. That's why it is a 2-deoxyribose. And then there is a phosphate group, that's it. These are the things. And the same thing, here I have mentioned, the same thing. And now what we are doing in this particular lecture? We need to study basic structure of purine and structure of adenine and guanine, basic structure of pyrimidine, structure of cytosine, uracil, thymine, and ribose structure, 2-deoxyribose structure, and then phosphate group structure. Individual structures we will study, that's it. With that, this lecture will be completed. It is mainly completely based on the chemistry only, and these structures, better please try to by heart this, try to practice this, try to remember this, because again they are needed in molecular basis of inheritance in biology. Not only this, even in the chemistry also you are studying the structure. So anyhow you need to remember. So better practice all these things. Clear? Now in the nucleotide, first we are talking about nitrogenous base. Simply I am writing N base, it is nothing but nitrogenous base. Nitrogenous base, in that first we are talking about the purines. So now first we will see about the purine structure. What is this purine structure? First we will focus on the purine structure. What actually the purine structure is? Purine will have actually two rings. Actually these are heterocyclic rings. Nitrogenous bases are heterocyclic rings. Why they are called as heterocyclic rings? Not only carbon, see these are the carbon positions. Not only carbons in the ring, even other compounds are also present. Hetero means different, cyclic. So there are cyclic ring, but it is not made up of only carbons. Here different, not only carbon, some other type of elements are also present. Here along with the carbon, nitrogen is also present in that ring. That's why it is called heterocyclic rings. This is how it is present. And now there is another one. It is like this. Understood or not? So this is how it is present. And in this, double bonds are present at these locations. And now this complete structure is called as the purine. This complete structure is called as the purine structure. So in the purine, originally if you see, there are two rings. This is one heterocyclic ring and this is another heterocyclic ring. Two rings are there. So purine is actually formed by fusion of two rings. What is the first side? Left side ring is called as, this left side ring itself is called pyrimidine ring. Pyrimidine ring. Please remember, if you understand this, after completion of purines, we will study about pyrimidine. At that time, this part we will not draw. We will draw only this. So you can easily understand this. So in the purine itself, one is a pyrimidine ring, other one is imidazole ring. Other one is imidazole ring. In this way, pyrimidine ring, imidazole ring. Both of them are heterocyclic rings. They are fused to form purine structure. This is what the basic structure of the purine. In this basic structure of the purine, we need to give the numbering. How exactly we are giving the number? This is the 1, 2, 3, 4, 5 and 6. This is 7, 8 and 9. This is what the basic structure of the purine with the numbering. And what are the positions of the nitrogen in this basic purine ring? At the first position it is present, third position nitrogen is present, seventh position nitrogen is present, and at the ninth position nitrogen is present. So these are the positions at which nitrogens are present. You should compulsorily remember this basic structure. And once you know the basic structure, here what happens? Adenine and guanine are substituted purines. So they will substitute some of these particular atoms or the groups so that you can simply add that over. So this is what about the basic purine ring structure. Clear? Now in this basic purine ring structure, now we need to talk about adenine and guanine. Adenine and guanine. Adenine is indicated with A and guanine is indicated with G. Guanine is indicated with G. Yes or no? So how exactly we are changing this? For that, once again I will draw the purine ring structure. So this is the purine ring structure. This is the basic structure of the purine. This is 1, 2, 3, 4, 5, 6. This is 7, 8 and 9. Same structure. In the previous slide, whatever I wrote, same thing I have written here. How exactly we are changing this? When you are writing the adenine structure, draw it as it is. As it is, without any change. Without any change, you just draw it. Even double bonds also you can write as it is. As it is, you have written this. And now here what we are doing? What is this position number here? This is the 6th carbon position, right? Now here what happens? On this carbon 1, 2, 3, only 3 bonds we are mentioning. That means what is this 4th one is present? Originally 4th one is hydrogen is present. Now this hydrogen is substituted with amino group. That means in place of hydrogen, amino group will come. In place of hydrogen, amino group will come. That's why it is called 6-amino purine. So adenine is nothing but 6-amino purine. That's it. So if you know the basic structure of purine, you can easily write the structure of adenine. Over. This is as it is. Only at this location you just add an H2. That's it. Over. This is the structure of adenine. and it is indicated with A. And where this adenine is present? Adenine is present in both DNA and RNA. Adenine we can find in both DNA and RNA. Clear. Next one, come to the guanine. For the guanine also, let us draw the basic structure, but let us not draw the double bonds. There is an adjustment of double bonds. So first we will draw the basic structure without double bonds. There is a basic, this is the basic structure without double bonds. Now, what are the positions that are affected? Sixth one, and this is the second position. Yes or no? This is the second and this is the sixth, right? Yes, these are the two positions. And now here what happens? Here at the sixth NH2 is there, but here at the second NH2 will come. Understood or not? And at the sixth, double bond O will come. At the sixth, double bond O will come. Now we just adjust the double bonds. For the double bonds, here you can keep double bond, here you can keep double bond. And the thing is, here also you can keep the double bond. But here you cannot keep double bond. Please remember, this is the change. Here you cannot keep double bond because here carbon already has 1, 2, 3, 4 bonds. So don't keep the double bond. If you are not keeping double bond, what happens? This N will automatically become NH. Even though you are not writing this H, even though you are not writing this H, no problem at all. You are not mentioning means by default hydrogen will be present. So you can mention or you can leave it, no problem. But don't keep double bond here. That's it. This is the structure of the guanine. So how we are writing this one? It is the 2 amino 6 oxy purine. 2 amino 6 oxy purine. Please remember, this is not IUPAC nomenclature. It is not IUPAC nomenclature. So don't worry, anyhow you can write 2 amino 6 oxy purine. 2 amino 6 oxy purine. That's it. This is what about adenine and guanine. It is indicated with a G, and this is also present in both DNA and RNA. So both adenine and guanine, both the purines are present in both DNA and RNA. In both of them it is present. This is what. With this, purine is completed, adenine and guanine is also completed. Clear? With this, purine's basic structure completed, adenine completed, guanine completed. So now we need to go for pyrimidines. We will draw the basic structure of pyrimidine. Slightly I will change this so that we will write all these three structures. That's it, very simple. Okay? Now we are going with the pyrimidines. You need to practice this. While you are observing this, you may think that it is very simple. But when you are start practicing this, then you will know the real difficulty. Okay? So please start practicing this. Pyrimidine. In the pyrimidine, what we are getting? Only the first ring. Only the first ring is called as pyrimidine ring. So draw only the first ring. Draw only the first ring. This is what. And here also double bonds. You just keep like this. Over. But the point here is numbering. In the previous one, if you are observing the numbering, we started here and we went like anti-clockwise. But here it is not like this. Here numbering will start with this. Numbering will start with this N, and we are going with clockwise. This numbering is very important in many books. In many books, they have given this number in a wrong way. They are still using this particular sequence only. That is wrong. This is only for the purine. For the pyrimidine, you should give numbering like this only. This is the standard. So please follow the standard. Don't make any mistakes here. Okay? This is what the pyrimidine ring. Single ring is there. This is also heterocyclic ring. So that's why whether purines or pyrimidines, whatever it is, they are heterocyclic rings. Clear? Now what we need to do? Slightly we need to change it so that we need to write cytosine. We need to write cytosine. We need to write uracil. We need to write uracil. And then we need to write thymine. We need to write thymine. These are the three things. First, what we will do? We will write all these without double bonds. First, we will write without the double bonds. Okay? This is the cytosine. And now let us see here in all these cases. Here also I will draw it without double bonds. Here also I will draw it without double bonds. First, we will draw without double bonds. But if you are observing very carefully, in all these cases, these two positions are affected. These are the two positions that are affected. Observe carefully. Here also, here only one. Here also these two. But the numbering in purines is different. That's why 2 and 6. But here, what is the numbering here? What is this position and this position? 2 and 4. This is 2, this is 4. This is 2, this is 4. This is 2, this is 4. Observe carefully how exactly we are trying to write this one. It will be very simple for us. See, actually, if you know the DNA double helical structure, in one strand if adenine is present, opposite strand thymine will be there. In one strand if guanine is present, opposite strand cytosine will be there. Guanine, cytosine opposite. Remember this. In the guanine structure, you just see these are the two positions having one amino and one oxy. Numbers are different, but here also positions are those two only. So if you remember, for guanine and cytosine, it is opposite. What is opposite? Here O is there, here NH2 is there. For the cytosine, it will go reverse. Go reverse means what? Here NH2 will come and here double bond O will come. That's it. Now you can keep the double bonds. So what happens? We can keep double bond here, we can keep double bond here. But here already carbon has 4 bonds, so we cannot keep double bond. So leave it. If you are leaving, what happens? By default, hydrogen will attach to nitrogen. Whether you are writing it or you are leaving this, no problem at all. It will be normal. No problem. This is what about the cytosine. So for this, how you will write? 2-oxy 4-amino 2-oxy 4-amino pyrimidine. 2-oxy 4-amino pyrimidine. This is what the name of this cytosine. And cytosine is present in both DNA and RNA. Cytosine is present in both DNA and RNA. Now we will go with the uracil. In the uracil, what happens? These are the two positions. These are the two positions. At both the positions, double bond O is present. At both the positions, double bond O is present. So you cannot keep double bond here, you cannot keep double bond here. You can keep double bond only here. That's it. So how we will write this? 2,4 dioxy pyrimidine. 2,4 dioxy pyrimidine. That's it. Very simple. So here automatically this nitrogen will get hydrogen, this nitrogen will get hydrogen. That's it. That adjustment we need to do it. That's it. So in the case of thymine, what we are doing? Whatever we wrote for uracil, as it is, exactly as it is. Exactly as it is means here double bond O, here double bond O. And here there is a bond. As it is uracil, you just write. But we need to add one more thing. It is as it is uracil, but we need to add one more. What is that? At this position. What is this position? Fifth carbon. At fifth carbon, what will be there? CH3 methyl is present. As it is uracil, but at the fifth position, methyl will be there. So how we are writing this? 5-methyl 2,4 dioxy 2,4 dioxy pyrimidine. 5-methyl 2,4 dioxy pyrimidine. This is 2,4 dioxy pyrimidine. That's it. Okay. This is what we are writing. And the same thing you can also write it as 2,4 dioxypyrimidine. You can write it as uracil. 2,4 dioxypyrimidine as uracil. So how you can write this? 5-methyl and this portion is uracil. 5-methyl uracil. This is the previous NEET examination question. So thymine is 2-methyl uracil, 3-methyl uracil, 4-methyl uracil, 5-methyl uracil. Like this, they have asked in the chemistry. So if you know this, there also you can write it. So that's why these are all like double advantages. So if you are studying in chemistry, you can use this concept in biology. If you are studying in the biology, you can use this concept in the chemistry also. This is what the basic information is. That's it. So in this way, pyrimidine, this is the basic pyrimidine structure. This is the cytosine, uracil, thymine. And the next important point is, see, in the previous one, what we studied, adenine and guanine. Adenine present in both DNA and RNA. Guanine present in both DNA and RNA. And in the pyrimidines, cytosine is present in both DNA and RNA. But uracil is present only in the RNA. It is not present in DNA. Whereas thymine is present only in the DNA. It is not present in the RNA. This is another important point that you need to remember. So in this way, we have completed the structures of this pyrimidine. Purine completed, adenine completed, guanine completed. Pyrimidine basic structure completed, cytosine completed, uracil completed, thymine completed. That means all the structures of these nitrogenous bases we have completed. Please practice. While you are looking at the board, it will be simple. But please practice. You need to remember them, because in the next lecture, deliberately why I am stopping after phosphate, because in the next lecture we need to interlink this. So when interlinking, I will directly write the structures. If you don't know what is that structure, then how you will understand the concept? So that is the reason why first I am giving the structures and I will stop it so that you can practice it. So when you are going for the next lecture, by that time you should be ready with the structures. So that what is the structure I am drawing, that you can easily understand. Clear? This is what. Now we are going for the sugar. In this sugar, first we are going with ribose and then we will go with deoxyribose. The advantage is, if you know the structure of ribose, you can easily write the structure of deoxyribose. So in this way, first we will study about the structure of ribose. So then we will go with the structure of deoxyribose. Ribose structure. Ribose. And please remember how I will teach you is, first I will give the ribose structures. Okay, different structures are in different way. Structure will remain same only, but in a different way we are representing all the information. I will show it here. And then what I will do is, in the same structure, I will do the changes. So you will get the 2-deoxyribose. So you can easily understand the concept. Clear? So first regarding the ribose. What is this ribose? Ribose is a 5-carbon containing sugar. Ribose is 5-carbon containing sugar. That's why it is a pentose sugar. It is a pentose sugar. Penta means what? 5 carbon containing sugar. That's why it is called as pentose sugar. So this is a monosaccharide. So generally these monosaccharides, they are having either aldehyde group or ketone group. So carbohydrates, these monosaccharides, generally how we will define, they are the polyhydroxy. Many hydroxyl groups are present. So polyhydroxy, either aldehydes or ketones. Now the question is, this ribose sugar contains aldehyde group or ketone group, that you need to tell me. So what type of sugar it is? Ribose sugar is aldehyde containing or ketone containing? It is aldehyde containing. Aldehyde group is present. That's why ribose is aldose sugar. 5-carbon containing, that's why it is pentose sugar. Aldehyde group containing, that's why it is aldose sugar. Understood or not? And what is the basic formula of this one? It will follow the normal basic formula of monosaccharides. What is the normal formula of the monosaccharides? CN, H2N, ON. Yes or no? In this N, what is the number of carbons? 5 carbons. So here it will follow same formula, C5, H10, O5. So ribose sugar is following the basic formula of the monosaccharides. Yes, it is following basic information. Now in this, how many carbons are there? 5. 1, 2, 3, 4, 5 carbons are present. Clear? Now, out of these carbons, aldehyde group. Aldose means what? Aldehyde group containing sugar. So aldehyde group means always aldehyde group will get the first carbon. This is the first carbon. This is second carbon, third carbon, fourth carbon. This is the fifth carbon. In all these sugars, if you are observing, the last one will be CH2OH. Last one is CH2OH. Clear? Now, for second, third, and fourth carbons, for these 3 carbons, how exactly these hydrogens and hydroxyl groups are present? In the case of ribose sugar, on the right side, OH group is present. On the right side, OH group is present at second, third, and even fourth carbon also. So that on the left side, hydrogen is present at all the 3 carbons. Observe the structure carefully. In the case of ribose sugar, first carbon CHO, last carbon CH2OH. For second, third, fourth carbons, on the right side OH is present, on the left side hydrogen is present. This is, if you are changing these, different structures will get. So you might have studied about isomers concept, right? So here this is how the ribose sugar is present. And now this ribose sugar, where this is the last chiral carbon. Already in the previous lecture itself, I told you about chiral carbon. When we studied about amino acids, I told you a carbon that is attached to 4 different types of groups is called chiral carbon. In this case, 3 chiral carbons are present. So here, on the last chiral carbon, OH group is present on right side. On the last chiral carbon, OH group is present on right side. So on the last chiral carbon, if OH is present on right side, it is called as capital D isomer. If you don't know about the isomerism concept, please consult the chemistry faculty. They will explain you much better. So please remember, on the last chiral carbon, if OH is present on right side, it is capital D type of isomer. If it is suppose reverse, OH is present on left side, then it will be capital L. Capital D, capital L isomerism is different from small d, small l. Small d, small l is also called as a plus minus, and that is completely different. Many students are not getting the difference between capital D, capital L and small d, small l. They are thinking that both of them are same. If you are still thinking that both of them are same, your concept is wrong. So please get that concept clear first. Okay. Here, here we are talking about capital D isomer, and here OH group on last chiral carbon is present on right side. That's why it is the capital D isomer. That's why it is capital D ribose. It is capital D ribose. This is what the structure of ribose sugar. But generally, see, this is present inside the cell. We are talking about this ribose as part of RNA molecule, and that RNA molecule is present inside our cells. Inside our cells, water is present. That means ultimately this sugar is present inside the water. So in the solutions, see, this structure is called a linear structure. Generally in the solutions, it is not present as a linear structure. It is present as a ring form, cyclical form. Cyclical ring form it is present. The same thing I am converting into a ring form. So how you are able to convert this into a ring form? Let us see this. 5 carbons. Last one is not affected. So write as it is. Second, third also not affected. So write as it is. Second, third also not affected. Write as it is. OH group of last carbon. OH group of last chiral carbon will interact with carbonyl carbon. So that now this OH will rearrange like this, and on this carbon what happens? Here you will get two options. Please remember, these are also anomers. They are called as anomers. ANOMERS, anomers. On the carbonyl carbon, I will get two options. What is the first option? On the right side, if I am writing OH, left side I can write H. Second option. On the left side, if I am writing OH, on the right side I need to write the H. Both the options are there. So now what is this option? Here on the left side OH is present. That's why it is called as beta form, beta anomeric form. If you don't know anomeric concept, so please consult with the chemistry faculty. So please understand these. These are very important. In the biology, they will not ask about whether capital D or beta. But in the chemistry, definitely for NEET examination, you need to know these. You need to know about these anomers. You need to know about capital D, capital isomers. Compulsory. It is compulsory. In the biology, we are not teaching all these things, but in the chemistry, you need to know this. If you are having any doubt, so whenever with this particular concept they are teaching, so please ask them. Please make it clarify. So here what happens? OH on left side, H on right side, it is beta form. Suppose reverse, OH on the right side and H on the left side, suppose. Then what happens? That will be the alpha form. But in the case of RNA, what type of form is present? Beta form is present. Because of this, it is beta. And here actually on the right side, OH is there, right? So that's why it is capital D. Beta capital D ribose. Beta capital D ribose. Understood or not? So in this way it is present. But whenever we are writing the complete structure of DNA or RNA, writing like this will be highly difficult. So that is the reason why the same content we are representing in a Haworth's projection. These are called as a Fisher projection. But instead of this, we are writing in Haworth's projection. Haworth's projection. Haworth's projection. In the chemistry, you will study about all these things. So please observe carefully. So now when we are representing this ribose structure in the Haworth's projection, what type of ring we are using? We are using a furan ring, Furan, furan. Furan ring. What is this furan ring? Furan ring is how many carbon containing? 1, 2, 3, 4. 4 carbon containing. Furan is 4 carbon containing. But members, if you count 1, 2, 3, 4, 5, oxygen is also considered as a member. So 4 carbon, 5 membered ring. 4 carbon 5 membered ring. You need to understand all these things. In the chemistry, definitely you will study this. So at least you please try to understand what I am saying. In the biology, even though they are not much important, if you by heart this particular structure, that is enough in biology. But try to understand the concept here. So this furan ring we are using, furan ring is having 4 carbons, 5 membered ring. Clear? Now. But here total how many carbons are there? 5 carbons are there. 1, 2, 3, 4, 5 carbons are there. But inside the ring, only 4 carbons are there. Then how you will represent this? And here we are representing, this is the first carbon, this is the second carbon, this is the third carbon, this is the fourth carbon. Then where we will represent the fifth carbon? Fifth carbon is projected from this, and this is how the fifth one is represented. And then this information you need to transfer here. How you will transfer? Observe carefully. In this structure, those which are present on the right side, those which are present on the right side, they are represented above the plane. They are represented above the plane. Those which are present on the left side, they are represented below the plane. Understood or not? This is how we are representing. So first let me represent the right side. Right side information means above the plane. First carbon right side H. First carbon right side H. So it is above the plane. Next second carbon right side OH. So second carbon OH above the plane. Third carbon OH above the plane. Right side. So it is how you are representing. Understand this. Here on the right side. Here above the plane. Now we will go for this one. So here left side means below the plane. First carbon OH below. First carbon OH below. For second carbon H below the plane. Third carbon H below the plane. Understood or not? Next one. Fourth carbon. Fourth carbon. Sorry. So this will be like this. So here we have changed it. Sorry. I am very sorry here. So we have changed it. This is the first carbon. Second carbon. Third carbon. Fourth carbon, fifth carbon. Up to this it is fine. So this information we need to turn like this. This information we need to turn like this. So those which are present on the right side, they will come below the plane. This hydrogen will come below the plane. This is say for second carbon right side. So this will come below the plane. Third carbon right side, it will come below the plane. Sorry. Okay. So this is what happening here. Next one, those which are present on left side. So since you are turning like this, so left side information will come above here. So this OH will come above for first carbon. For second carbon above, hydrogen will come. Third carbon above, hydrogen will come. Fourth carbon, here already O is mentioned here. So hydrogen you are writing like this. Then what about the fifth one? CH2OH, right, as it is here. CH2OH. Okay, so we made a small mistake. I am very sorry. So please remember, so in this representation, whatever they are present on the right side, here they should be represented below the plane. Here whatever they are present on the left side, they are represented above the plane. But mainly anyhow 4 is connected to 1, so that's why this Y is there. So for these two we are not worried, but for 1, 2, 3 you should be very much careful. For 1, 2, 3 you should be very much careful. This is how we are representing the information. So all the 3 are correct only. But from the next lecture onwards, whenever we are studying about the structure of ribose, directly we will write like this. Whenever we are writing the structure of ribose, directly we will write like this, because this is easy. When we are writing about not just ribose sugar, nitrogenous base plus ribose sugar plus phosphate, these many we need to link. So this structure is easy. And this is the Haworth's projection. And here we are using the furan ring. Understood or not? So in this way we are representing the information. So this is the ribose structure. So even though these two are also correct, but generally we are using the ring structure like this. In this ring structure, wherever hydrogens are there, you don't need to mention them. So don't keep any line. So simply you just leave it. Don't draw the line. Sometimes what they are doing is they are making a line and then they are leaving like this. Suppose I will give you one example. Suppose you are making a line and then you are leaving. Making a line and leaving means it is not a hydrogen. In the organic chemistry, you might have studied about this. You are making a line and leaving means what? Here CH3 will come. So this will be a wrong. So if you don't want to draw this, what you need to do? You just simply leave it like this. Simply leave it like this. Simply leave it like this. So if you are leaving means by default hydrogen will come. But if you are making a line and leaving means methyl group will come. So both of them are different. So that's why please observe carefully. So if you want to write hydrogen, make a line and write hydrogen. If you don't want to write hydrogen, don't draw the line at all. Don't draw the line at all. Simply leave it. So if you are not drawing this line, so simply draw OH on the above, don't draw anything. Still it is correct. So in this way we are drawing the structure for this ribose sugar. So with this the structure of ribose sugar is completed. And now what I will do is slightly I will change this particular one so that you will get the structure of 2-deoxyribose that is present in the DNA. Ribose sugar is present only in RNA. 2-deoxyribose is present in DNA. So how we are writing that particular structure? Observe carefully. Now within this one only I will change it. Now we need to go for 2-deoxy. At the second carbon, D means what? D means what? Removal. Removal of what? Oxygen. At the second carbon, oxygen is removed. Where? Here. So here what happens? Here instead of OH, what will come? Hydrogen will come. This is what? This is what will be. This will be 2-deoxy. Capital D, 2-deoxyribose. Instead of ribose, it is 2-deoxyribose. And here what we will do? At the second carbon, instead of OH, we are writing H. So what is the name of this one? Beta, capital D, 2-deoxyribose. Over. Remaining structure will remain same only. And here how you will change it? At the second carbon, instead of OH, you are writing the hydrogen. And please remember, if you don't want to draw hydrogen, don't draw the line at all. Leave it. Still it is correct. This is how you are writing. So in this way 2-deoxyribose, this is also 5 carbon, pentose sugar, aldose sugar. But here this is the basic structure for basic formula for monosaccharide. But in this case what happens in the overall structure, instead of OH, H is coming. That means H is okay, but O is missing here. So C5 is okay. H10 is okay, but instead of O5 it will be O4. So for 2-deoxyribose C5H10, O4 is the molecular formula. It is not O5, it is the O4. But remaining structure will be like this. So if you know the structure for the ribose sugar, you can easily write the structure for 2-deoxyribose. So this is how from the next lecture onwards, if you want to write the deoxyribose, 2-deoxyribose, this is how we need to write the structure. Clear? With this regarding the sugar also it is completed. And now the last one, that is regarding the phosphate group. Phosphate group. You can call this as a phosphate and you can also call this as phosphoric acid. Phosphoric acid. Which phosphoric acid? Orthophosphoric acid. Orthophosphoric acid. How you will write this? Phosphoric acid. H3PO4. Please remember, many students what they are doing is H3P small O4 they are writing. Can you write like this? H means it is hydrogen. Capital P small o, capital P small o, it will become polonium. It will become polonium. So this is wrong. So what you need? Capital H, subscript 3, capital P, capital O, 4. Now it will be hydrogen, this is phosphorus, this is oxygen. So you need to know how to write also. Many students instead of writing capital O, they are writing small o, that is wrong. So you should not write like this. So please observe carefully. So it is H3PO4 like this. So if you want to write phosphate, one H plus you need to remove. If you are removing H plus, what happens? H2PO4 minus. Same thing. Anyhow you can write it. If you are writing for this P and its valency is 5, so it will be like this. It will be like this. Yes or no? P double bond O, OH, OH, OH. This is the H3PO4. From the same thing, if you are removing the H plus, what happens? It will be O minus. H plus removed, so it will be O minus. So H2PO4 minus. This is the phosphate or orthophosphoric acid. Anything you can say it. Understood or not? And because of this phosphate group only, negative charge to DNA, negative charge to RNA is due to this phosphate group only. That's what we are talking. So in this way, phosphate group is also present in nucleotide. So nucleotide, with this all the structures are completed. So nucleotide has three components: nitrogenous base, sugar, and a phosphate. Nitrogenous bases are two types: purines and pyrimidines. Purines are again two types: adenine and guanine. Pyrimidines are three types: cytosine, uracil, and thymine. Adenine is present in both DNA and RNA. Guanine is present in both DNA and RNA. Cytosine is present in both DNA and RNA. Uracil is present only in RNA. Thymine is present only in DNA. Next, sugars, two types: ribose, 2-deoxyribose. Ribose sugar is present only in RNA. 2-deoxyribose is present only in DNA. And phosphate. Phosphate is present in both DNA and RNA. Understood or not? So with this, individual structures we have studied. And please remember, in NCERT very clearly they mentioned these nitrogenous bases are heterocyclic rings. They are heterocyclic rings, very, very important one. And with this, these structures are completed. Clear? So please practice all these structures so that in the next lecture I will try to combine these molecules so that different names will come out. And all these names were mentioned in NCERT, even in class 11th, even in class 12th also. So you need to compulsorily know these names with structures. Then only you can understand the structure of DNA very clearly. So please remember all these things. Clear? With this lecture is completed. We will meet in the next lecture. Thank you so much.